Yeah. Hi, good morning. So today there are few important announcements to do regarding few playlists and series that I'm going to start with this you know 11th and 12th biology lessons. Okay. Let everyone come so we can start. Yesterday, question 87. Option A is correct. Okay. So today and tomorrow we're going to discuss about this lesson, principles of inheritance and variation. Though in this school we separated the CBSE syllabus for your batch as botany and zoology, and we shared this lesson, uh, one third in botany, three-fourth in zoo. So this one fourth of botany is placed today. So that's why I gave only one day for botany, that is principles of inheritance and variation. It means three laws, and then their examples are quite enough. One day is more than enough when it comes to botany, right? So immediately you have to finish the botany and switch to zoology topics, right? So it can be a continuation also, because the same lesson you're reading, you can just keep reading, continue botany and zoology. Good morning. Good morning, Abhiniti. Good morning, Arshia, Akshya, Jeena Priya. Good morning, and Arshita Priya. Good morning. Okay, fine. So tomorrow we'll finish this entire lesson, because geology part of this lesson is quite elaborative, and more concepts will be there. Examples, chromosomal disorders, Mendelian disorders, and then sex determination systems. Right? We have studied very well in the case of academics itself. We did a lot of pedigree also, and unfortunately few members who really like to answer the pedigree questions, they are quite disappointed that the board CBSE board didn't ask pedigree. And yeah, last year also they didn't ask any question about pedigree, but they were asked in practicals. Okay, and at the same time for NEET, these questions would be, concepts would be important, important, so no need to get disappointed. Okay. Yeah, so the important announcement is nothing but I'm going to start a series of videos. Okay, it means for example, if this principles of inheritance and variation for your session, that is this is quick review session, right? So quick outline session, I'm taking 1 hour. Within 1 hour I'm trying to cover one or two lessons at outlines and important highlights, right? So you can, whoever already prepared all on these chapters, you may get, okay, these are the things I have to focus. But still, what I felt was like this 1 hour session is not it can be elaborated. So what I thought was like, I will post few series of or I will come live, or I will post few series of videos, okay, not in class time. Okay, you can go back and you can refer those outline, 1 hour outline for one chapter. So for one chapter, 1 hour, I will deeply I will discuss. So that is just outline. So this is one type of video I'm going to upload for every chapter. 38 chapters of biology, okay, both 11th and 12th. So it would be very, very useful for you guys. At the same time, the second type of video what I'm going to upload is that entire lesson. For example, if you take this principles of inheritance and variation, they were almost what do you say, six subtopics are there: Mendelian inheritance, inheritance of one gene, inheritance of two genes, sex determination, mutation, and genetic disorders. There are 6 parts within this lesson: 5.1, 5.2, 5.3, 5.4. Like this you can make use of these videos at any point of time. Okay, this is my plan, so that whatever the planned physiology that you're going to come, right? So 11th standard portions, so that you can get more benefit of these videos, so that you no need to waste the preparation time. Schedule time will not be disturbed, especially in the NEET schedule what I gave here. So that schedule will not be disturbed. So whenever you find 1 hour, 2 hours time and you're reading that topic, and when you feel you stuck, you can go through that videos and you can play n number of times also, so that you can understand. You can reach me at any point of time. So these are the series I'm going to upload very soon. Every day one video I will be uploading, both 11th and 12th standard video. Especially plant physiology is my target, because I know the understanding level. So I just wanted to make sure that you should not lose anything, as simple as it. Okay, let's move to today's topic. Okay, so please do consider subscribing and hit that notification bell, so that you will get instant notifications. So okay, let's move to the today's lesson discussion, that is principles of inheritance and variation. Is that my series is understandable, what I've told, right? Did you get it? Did you understand what I told, ma? One is full chapter videos will be there, and outline will be there in the same lesson. So you will have this live session, quick outline session, a detailed outline session, at the same time full chapter videos. Because I am teaching for almost 1 or 2 years for you, the botany aspects also I will cover, both 11th and 12th standard, so that it will be more, you can get access of this video at any point of time. Okay, fine. Okay, so let's move into this chapter, that is principles of inheritance and variation, right? So if you say this principles of inheritance and variation, what we have to focus more here. So what we have to focus is the definitions, right? You should not get confused with the when it comes to homozygous, heterozygous. These are very simple examples. When we simple definitions: homozygous and heterozygous condition, recessive character, dominant character. Good morning, Sridaksha. Right? So there are traits, alleles, right? So these are the things which you have to just brush up. Okay, no one will ask any question based on these definitions, but still, right, if they ask correct or incorrect statement, you should not lose marks. So you should be extremely careful about it. So whoever not attending these classes, again I'm telling, they will miss some aspects that we are covering, highlights, etc. So make sure your friends are attending the class. Just inform them as well. Okay. So what is an important aspect of this lesson is that the principles, the laws actually. So what is the inheritance laws we have, and then variations, right? So how do variations were created? So when it comes to Mendel laws, right? So you have 7 characteristics that why the Mendel opted, right? So if you think Mendel has got this probability right, but not others. Why is that he applied this statistics to this this characteristic 7 traits, and more importantly, there is no linkage in these characteristics. That's what he is able to observe this probability patterns over n number of generation in different traits, almost 7 different traits. Okay, so this is one of the aspects. That's okay. So that's what the selection of plant is. What one of the important aspect that Mendel has done. Okay. Right. So coming to the next one, that is inheritance of one gene. So if you take this gene, right? Gene is a DNA sequence that Mendel has no idea, and he called factor, blah blah blah. Means like these are all things we already covered in academics, but we need to once just quickly go through that. Okay, who gave name gene, and then what is a gene, and DNA sequence, and etc. That how this single gene is able to actually inherit, what might be the patterns in it. So here in one gene inheritance, or single gene inheritance, or double gene inheritance, two gene inheritance, you need to more focus on the ratios. Okay, phenotypic ratio, genotypic ratios, right? Back cross, test cross ratios. So ratios are very very important in this, and they may ask you in a jumble manner or in correct or correct. Okay, from below, then the below given ratios, which is back cross, which is test cross, which is what are the ratios of genotypic ratio of this character, and these are the things you need to focus on. Okay, so much not about the theory aspects, but you need to focus on the ratios of each and every concept here. So for example, what is there, you take you know a single gene inheritance. If you are crossing between a homozygous pure lines tall and short, what is a phenotypic ratio, what is a genotypic ratio? If you do back cross, and what is and if you do test cross. So in the back cross, crossing hybrid with its any parent. So these are the statements very important. I already gave it in academics class as well. Okay, I mean for 11th standard, your juniors. So this is the first aspects. So these are the first these are the initial aspects that you need to really understand, so that you can when you read this entire lesson, it will make more sense. So when we talk about back cross is something crossing of hybrid with its any parent. Hybrid in the sense like F1 generation. So pure lines and then F1 generation. So F1 generation is again crossed with any one of the parents is called back cross. And if we cross this same hybrid, F1 generation hybrid, with the recessive plant, it is called test cross. So we have to see that in monohybrid cross, test cross ratios would be 1:1 ratio. And then these ratios are very important that you need to focus. Okay, so when we talk about principles, right? So there are different principles that Mendel came up with. That is, one is first law, there is law of difference, and then second is law of segregation, right? And then we within this one gene inheritance, we talk about first and second law, that is law of dominance and law of segregation. When we talk about two genes, we talk about independent assortment, right? So that is a third law of Mendel. So this is very interesting results that Mendel got, and everyone got shocked. But still, as an many of the scientists and general population really couldn't take this or understand the math behind this Mendel process, and he didn't get credited when he published all this data. But later, again, these works were again taken up by scientists, and they proved that yes, this is a pattern that you see in this monohybrid and hybrid cross. And then everyone started following or believing or in this concepts, the Mendel laws, right? So these are the two laws. So the law of inheritance, right? In this laws of inheritance, you have got law of dominance and segregation. So these definitions are there, right? So what actually makes a law of dominance dominant? In the sense, like, what are the features of this dominance? So these are the areas where you may get correct or incorrect statement. So this is a where this is the type of, these are the type of questions where we may go wrong. So suddenly we omit this not or incorrect. So suddenly in with the rapid speed, we assume, we think that by overlooking, we think that this is correct answer. So you think that the statement, the question given is to pick the correct answer, but you will be leaving or you will be overlooked. So that you should be very much careful about. Read question twice, right, and then it would be useful. So every Thursday I'll give you some tips and tricks. Okay, during our doubt session, tips and tricks will be given. Okay, how to handle any question and type of model questions, etc. will be discussed, right? So when it comes to law of dominance, what are the aspects of the law of dominance? For that matter, okay, you guys are following, I guess. Okay. So, for example, uh, uh, the features that are present in the law of dominance and then segregation, you need to thoroughly read, and then you should be identifying in questions if they are given incorrect or correct statements. For example, factors occurs in pairs, and then these characters are controlled by discrete units called factors. So these are the points that you come across in dominance, law of dominance. And in segregation, segregation, and what exactly segregating is that this chromosomes during the gamete formation, right? So when you talk about alleles, right, homologous chromosomes and presence of this alleles and their segregation is what we talk in law of segregation. So these statements you need to go through once, many times, so that you will not go wrong in these statements and definitions, right? And they have given simple crosses that your favorite cross, that is round and yellow, this cross, and then the ratios that we got that we get phenotypic, both phenotypic and genotypic ratios you need to remember. Okay, so in this genotypic ratio is dihybrid cross. When we talk about inheritance of two genes, that is the dihybrid cross is important. Okay, and in this dihybrid cross, the genotypic ratios and phenotypic ratios are important. So you should be remembering this 1:2:1:2:1:2:1:2:1. So this genotypic ratios and phenotypic ratio, that is 9:3:3:1. So within this 9 phenotypes, what are those genotypes you need to remember? Okay, this is where you may feel that you may miss marks. Like these are memory based actually, right? So when we talk about dominance, right, we have got different different types, right? If you see this concept of dominance, you will see that there would be alleles which are unmodified, alleles which are modified, right? So when they are modified alleles are there, what are the three types of functions they do, right? When the modified alleles, right, how what they are responsible of. So when the alleles are modified, what happens is, last class also I discussed, right? There would be a less efficient enzyme than the normal one. Okay, I told now this genes and the gene production proteins, and proteins or few will be enzymes, right? So the enzymes which we produce from the modified alleles, we are talking about concept of dominance. Modified alleles, there are three aspects that covered in NCERT. That is, when the allele is getting modified, it can show three different effects. One is less efficient than the normal one or normal, or a non-functional enzyme, or no enzyme at all. Okay, so these three aspects are important for correct or incorrect statements. Okay. So you need to remember. So if you see the other patterns of inheritance, you will be finding incomplete dominance, codominance, and multiple allelism. So they are a little deviated from Mendelian inheritance. Okay. So if you see the incomplete dominance and its genotypic ratio and its phenotypic ratio. So what you can do is you can make a tabular form while you are preparing today. You can write this incomplete dominant. You can write with dominance and then segregation. You can start with the and there phenotypic ratio, genotypic ratio, phenotypic ratio, genotypic ratio, and then you can put another three aspects, that is incomplete dominance, phenotypic ratio, genotypic ratio. Put the genotypic ratio first, And then codominance, genotypic ratio, phenotypic ratio, and multiple allelism. How this multiple allelism would occur, and how these three alleles would control the expression of these blood groups is what you need to identify. So they may give different combinations of the alleles, and they may ask you to pick the right blood group. Are you able to understand? Are you able to follow, guys? Oh, sorry, I didn't. Satyapriya, sir, can you please explain the difference between segregation of alleles and chromosomes? Satyapriya. Okay. Yeah. Yeah. Yeah, Satyapriya. So Satyapriya, she's asking, what is the difference between, you know, I didn't see the question actually, right now, just now I've seen segregation of what is alleles and versus chromosomes, right? If you see there's alleles, what is an allele first of all? What is an allele? Oh, Satya, this is Dharani. Yeah, Dharani. Okay. So what is an allele? Yeah, please respond. Please do respond, guys. What is an allele? What do you understand by the word allele? Yeah, two copies of gene, right? Might be an alternate form of the gene also. So alternate form of a trait or character, right? Yeah. If you see these alleles, where exactly they are present? Two copies. A pair of contrasting character of a gene. Yeah, okay. Correct. Yeah, please answer. Yeah, yeah, yeah. Pair of yeah, correct. Others, please do respond. Okay, let's see then. So if you see this, the difference between the question asked was the difference between the segregation of alleles and the chromosomes. So if you see here, I always take this analogy that if you take a bus, right? Bus is a chromosome and the seats are like a locus. In the locus, right, or loci, the persons who sits or maybe an alleles or genes. So gene has an alternate form, that is allele. So what happens is when the chromosomes segregate, along with the chromosome, even the alleles also get segregated. Okay. So during, if you see that during meiosis, right? During meiosis, what happens is this alleles will get segregated along with the homologous chromosome separation. So when you see these homologous chromosomes comes, and then in prophase one of meiosis one, what happens that there's all packaging in all these five phases occur and recombination occurs, right? So after which what happens is these alleles which are present on these homologous chromosomes, along with the chromosomes segregation, the chromosomes will move. If you see the further meiosis, if you see the meiosis 1, prophase 1, after prophase 1, metaphase P mat C, right? That is prophase, metaphase, anaphase, and telophase. During this M1 phase, that is meiosis 1 phase, what happens is the homologous chromosomes, which has got this alternate form alleles, also will get segregated along with the chromosomes. So when the chromosomes moves and segregate into 4 different gametes, these alleles also will go into individual cell. That's what we get the recombination also. What happens is one cell, one gamete at the end of meiosis 2, what happens is we find one type of allele in one type of gamete, one type of allele in one type of gamete. Likewise, we get 4 different types of gametes. So how do we produce these 4 different types of gametes? In both ways, right? In both ways, one is chromosome is actually moving into different cells. It is when it is segregating along with the chromosomes, even the alleles also get segregated, separated. This is how this separation of chromosomes do occur. Understood? So when we talk about difference between segregation of allele is nothing but it's not actually different. It's like same process, like within the same meiotic divisions, the alleles are moving along with the chromosomes. Okay, first they occur in pairs. So these are all similarities actually. So first they occur in pairs and they segregate individually. Yeah. So that's how chromosomes segregate, and that's how alleles also will get segregate. That's what the law of segregation is explaining. Okay, understood, right? Yeah. So the meiosis 1, meiosis 2, if you remember that, it will be more easy. So you can take any two alleles, like you can take 2 homologous chromosomes. Okay, that is one chromosome from paternal chromosome, one is maternal chromosome, and you have a capital or the trait that is, for example, red hair, and you have a black hair that is smaller. So what happens is these 2 homologous chromosomes are there, right? So these 2 would eventually enter into the next phases of the meiosis, and this allele will go into one gamete and this allele would go to other gamete, or they may get a compensation effect also. But this is how the alleles would actually separate during meiosis, and they segregate along with the chromosome. So here the capital R is there, that is red hair, and this is small r is there, right? So this is one cell. Okay. So when one cell is undergoing this meiosis, what happens is it undergoes 2 divisions, mitotic one, meiosis 1 and meiosis 2, and then results in formation of 4. So likewise, these alleles will get separated. Okay, let's move on. Okay, you understood, right? Yeah, great. So let's move on. Yeah, when we talk about polygenic inheritance, this polygenic inheritance is important, and the examples are important. And sometimes the skin color, if you see this skin color, I always do this, we use this example also, right? If you take a classroom and or any population you take and you see the skin color, right, it varies. It's not just dark and dark and light, but you will find the different combinations, right? Different percentages of variation. Intermediate skin color also you will be finding because of the melanin pigmentation and the etc. So you may get questions like, they may give the genes which are controlling the skin color, and they may ask which would form the darkest skin and the lightest skin and the intermediate skin. So you need to identify capital A, capital B, capital C. If you take that as an example, if they give like that, which causes with the homologous chromosomes, capital A, capital A, capital B, capital B, capital C, homologous chromosome from paternal and from maternal, from father and mother. you're getting both dominant, right? This is capital A, capital A, capital A, capital A, capital B, capital B, capital C, capital C. But if you get small a, small a, small b, small b, small c from both the parents, you may express the recessive traits. So then it will be more fair or more light in color. So that is the lightest and the darkest and the intermediate. Capital A, small a, capital B, small b, and capital C as small c is what the intermediate thing. So this polygenic inheritance and the genes and the products that what we get. It means the darkest skin color has got what genotype. So these are the aspects you need to focus. So polygenic inheritance and pleiotropy is more important because these are the concepts where they are little different from the Mendelian laws. Okay, because here if you see here three different genes are controlling single character, polygenic, right? Polygenes, many genes are controlling single character, that is skin color. Pleiotropy is opposite again, right? So here single gene exhibits multiple phenotypes. So one gene will be there, but multiple phenotypes will be there in pleiotropy. That's what these two things are. Two aspects are very important when it comes to what is a this non-Mendelian inheritance. Okay, these are other patterns of the inheritance. Okay. So what we discussed is we completed the botany aspect, right? That's what I told, right? This is only 1/3 or 1/4 of the lesson is covered in botany, and the two these are quite a memory based, but statements are very important because all the entire lesson definitions are comes from the first part. Okay, if you don't understand how this segregation happens, what are alleles, what are genes, what are factors, what are traits, what is genotype, what is phenotype, what is homologous and what is heterozygous, what are F1, F2 and F3 generations, and what is test cross, back cross. If you can't understand these things, and you can't this lesson will not make sense. Okay, the next aspects. Okay, when it comes to this chromosomal theory of inheritance, right, we are as per your batch, we are entering into the geology part, that is chromosomal inheritance of chromosomal theory of inheritance. So I told now this is a quick outline of the chapter, and again I will be posting a detailed outline and then the full chapter videos also. Okay, so please stay tuned and you see the playlists. Okay, so I will be keep updating it and I will be posting links as well. Okay. So let's see about the chromosomal theory of inheritance. So here we have to remember about the scientists' names and who gave this chromosomal theory of inheritance. What are the points that we cover in chromosomal theory of inheritance? It's a merge, right? If you see this chromosomes are vehicles of heredity. I told, right, I took an analogy of a bus. Right? How do they, along with the chromosome, the passengers are moving. So along with the chromosomes, even the alleles would get segregated once this chromosome, which are diploid or actually undergoing meiosis, reduction, division to become haploid. So when this chromosome content ploidy is changing, even the alleles also do segregate. That's actually how these alleles and chromosomes do segregate. Mostly they are all similar. Okay, that's all. So Sutton and Bovary gave this chromosomal theory of inheritance, right? And the theories, what is a theory suggests. So you may expect in statement wise, right? So chromosomes are not vehicles of heredity. So that is an incorrect statement. So you need to pick which are correct or incorrect statement from this theory. So you should not ignore at all. And then two identical chromosomes forms a homologous space, and homologous space segregate, right, during gamete formation. So these homologous chromosomes would segregate during gamete formation, and then independent pairs segregate independently of each other. For example, chromosome number 1 of homologous chromosomes would segregate separately, and they don't depend upon chromosome number 2. It means whether chromosome number 1 comes or 2, whether chromosome number 2 comes along with the 1, it doesn't really care. It just segregates individually, independently. So that's what this theory suggests, the chromosomal theory of inheritance. Pattern of inheritance, how they are actually segregating, right? So here you need to remember about the entire meiosis and the phases in meiosis. Meiosis 1 is a very critical thing, right? So this meiosis is something which is covered in cell division, right? Cell division chapter, right? Once we will go through and 11th standard topics we will take up. So quickly go through this once. That is meiosis 1. What are the different phases, right? P mat C, again prophase, metaphase, anaphase, telophase, right? Likewise, how do these chromosomes actually segregate? So this is what chromosomal theory of inheritance stated. The aspects, the behavior of genes are factors, or how do they segregate, is given by this, chromosomal theory of inheritance. So that's what when we talk about the separation or segregation of genes or alleles, right? They show similar pattern of segregation, similar behavior of moving into new gametes, new cell. When gamete formation takes place, why we are studying this, and where do you find this entire recombination, segregation, or everything? You find it in meiosis 1, and then meiosis 2 is nothing but it just as similar as mitosis, right? Which results in the end products of meiosis 1 is 2 cells, and then that further divides to form 4 cells. So along with the chromosome vehicles, even the genes or traits or alleles also will get segregated. So probably both behave the same way. And then we come to speak about the Thomas Hunt Morgan, and then we talked about the gene mapping, right? So when we talk about the centimorgans and how do we really map, and how we have done in academics itself the problems, numericals and other problems associated with concepts, right? So when we talk about the linkage, right? When we talk about the linkage factor, and why we see this Drosophila melanogaster is one of the favorite organism for studying the genetics in animal models, right? Insect models for that matter. The characters of this Drosophila and the characteristics of pea plant are important. So you need to brush up those things which are memory based. So let's move to the linkage concept, right? So are you able to follow, guys? Are you able to follow? Yeah. Yes. Okay. So we'll move further. So what about remaining? Are you able to follow? Yeah. So please be active. Today again, a few members came. That's it. I don't know what makes you skip the class, but still, 1 hour if you can't spend, how it will be helpful? I don't understand. Anyways, okay. So you're able to follow, right? Yeah. Okay, we'll continue the next aspect. So before that, again, I just wanted to make clear about this, you know, segregation and independent assortment. Independent assortment is something which we talk about chromosome behavior, right? How do these chromosomes are moving, right? How they are independently assorting, and law of segregation, which actually explains the differences between what do you say, uh, segregation of alleles and chromosomes. Okay, so if you see in what might be the difference, right? So we already talked about both the behavior is of similar kind, that is how do they separate and segregate, right? But when it comes to this you know uh, what do you say, the segregation and independent assortment, right? The alleles do segregate during gamete formation, right? So which would be established within the meiosis 1, right? Meiosis 1, it means metaphase 1 itself, they would be established. The segregation would the gametes, the alleles which needs to segregate, right, within this homologous chromosomes would be established by the end of meiosis 1, that is within the metaphase 1. But when we talk about law of independent assortment, we'll talk about genes on separate chromosomes assort into gametes independently. So they don't actually interact with the other genes until or unless they are linked genes. So this is the exception, right? So by the end of metaphase 1, right? So these dihybrid, this the genes which are controlling two traits, more than one trait, right? So they do independently assort. So that's what the difference between the law of segregation and law of independent assortment. Where in the law of segregation we more talk about the alleles, right? So here only only one gene is there, one character is actually they are controlling. But when it comes to dihybrid cross, it's single. There are more than one trait is observed here. So and they are not actually dependent upon their segregation is not dependent upon the other factors or other traits, so that they independently assort. These alleles will independently assort until or unless, or for example, exception is linkage. So when the genes are in linked, right, they do no, they do have an impact on the segregation process. So with this, we start this linkage concept. Why? What exactly the linkage is there? How does that affect the recombination and variation also? So here we take the model organism as Drosophila, and we'll be crossing two process we will take. And then here we need to observe the definitions of linkage and recombinations. What is linkage? I gave in academics and NEET classes also that how do we see this linkage, right? When the linkage is high, right? What is the distance between the two genes? So what exactly linkage is? Nothing but the distance between the genes, the physical distance, physical association of two or more genes, right, within a chromosome. So within the bus, right, within the bus you have the genes seated, right? So when they seated very closely and they are linked, right, their segregation would affect, right, their recombination would affect during the meiosis. So I gave it one kind of a equation I gave, right? So how the linkage is inversely proportional to recombination or directly proportional to recombination. So these are the aspects. They are quite a mnemonics that you can go through. So please go through the running notes as well. The linkage and recombination concepts are very important. So the process between the two types of process we see, one first cross is yellow bodied with white eyed females, crossed with brown bodied red eyed males, wild type, and then the second cross is white eyed mature wing. So you see this example, but what we have to focus here is the pattern. So which cross has got which, how much parental type is there, how many recombinants are found, what makes this recombination less. So what in NEET exam you may give A, B, C genes and they will be closely associated. A, B will be closely associated. B and C the distance would be more, right? So they may ask you that which genes shows the highest recombination, which genes will show lowest recombination. So in that way, you need to identify and apply this concept, or directly they may give this cross A, cross B diagrams in the NEET question itself, and they may ask you to find out which has more recombination types, percentage of recombination. So likewise, who gave this recombination frequency between gene pairs, right? So the Alfred, who is a student of this Morgan, and then he came up with this concept, right? The linkage, I mean the distance between how do we measure this distance, centimorgans, etc. So and then we come up with genetic maps in the Human Genome Project. So this is the applicatory part. So the scientist names within this linkage concepts are important. So the percentage recombination occurs when the linkage is high. When the linkage is low, what is the percentage of recombination, recombinants or parental types? Those percentages are important. There is a concept behind that. Linkage is important because they may not ask the same example in the NEET test. They may be asking a different example. I told, right, they may give A, B, C, D, E, and they may give distance also. 5 centimorgans and then 10 centimorgans, and they ask you to find out which has got more recombination, right? So we already dealt during the academics test, academic board exam as well when it comes to numericals, right? These are the aspects you need to cover in the linkage concept. These are the hotspots. Now when we talk about sex determination, right? So the sex determination, types of sex determination and their mechanisms. Examples are very important. You should not get confused with the human sex determination system with the other sex determination systems, right? I gave additional sex determination systems also which are not covered in NCERT, right? So people do ask, like students do ask a question that, sir, I'm covering entire NCERT, but still I'm not able to answer every question that was that was I'm encountering in different tests. So the problem is that it's not that this entire NCERT is a Bible, Quran, and then Bhagavad Gita, right? Yeah. So, but here what we need to see is that when we study a particular concept, we need to study even the the overall, the nearby concepts as well. So when we are in NEET class, I used to explain when we talk about cancer. I explained about the different aspects which are not covered even in NCERT, right? But it's not that whatever not whatever not it's sure that whatever not covered in NCERT they will not be asking, but based upon what is there in NCERT. For example, they give tuberculosis, but they don't talk about much about the tuberculosis. But that's our responsibility and that's our interest to cover at least what is a organism, what is the mode of transmission, right? These are not even extra points. These are the things which we need to even understand better about that concept, right? So this is where we lack, and that too, different test series will have a different approach, and they may take different syllabuses as consideration. So nothing to get panic, but if you are very sure about this NCERT textbook, maybe you can score 320 marks, 320 to 330, line by line, everything you are sure, and every concept you can apply. It's not that every line you can really memorize and all, no, but you have to even apply. You should understand the question itself, right? So this is where many students would lack, that sir, I'm studying entire NCERT top to bottom, but I'm unable to do, is that because few test series what you give, it will be based upon the other syllabus as well. They may take the different other than more than NCERT they may go. So you nothing to lose any confidence, right? You keep reading NCERT. On that basis I'm giving outline and I'm giving previous year's questions, at the same time practice questions. So whenever you don't find, you are unable to answer that questions, go back to that concept in different books or different study material, so that you will be more equipped. Here this and NEET test, or for that matter any for example IAS test, IAS exam, if you take that civils exam, there is no specific syllabus. If it is a geography, they talk about plate tectonics, like it will be vast. So they cover a lot. That's it. That's what, if you see an IAS officer, they are very much rich in their any public service preparations. Government employees or any other students, if you see that the public services who prepare for that, there will be vast knowledge will be there with them. Why? Because when we talk about sex determination, they cover other sex determination systems what are available also. Okay, NCERT is given only this, I will not study other things, means it don't fetch anything. That's what this extra mile you have to go, but only based upon the NCERT syllabus and the text were given. I hope I am clear, right? So when we talk about this section, so there is nothing to lose hope and don't get discouraged. Okay, you have time now. You please plan properly and ask doubts at any point of time. Okay. Okay. So the next aspect is when we talk about this. Sex determination, the types of mechanisms that we observe during this sex determination, right? We have XX, XY, XX, XY, ZZ, ZW, right? Their examples are utmost important. You should be writing like a tabular form, XX, XO and its organism, right? What is a peculiar point? Whether it is a male determining male heterogamity or female heterogamity. So these are the important examples, memory-based questions you will be getting, right? So especially the humans are important. At the same time, honeybee is something a tricky kind of an sex determination system, where they don't talk. The sex is not determined by the X chromosomes, but it is determined by the fertilized or unfertilized eggs. So that you need to focus on. Okay, guys. So tomorrow we'll be discussing about mutation, genetic disorders, and the next half of the lesson. So as of now, what I have gone through is still sex determination I covered. Sex determination, I am telling again, I didn't much talk about each and every sex determination here. I'm just giving, what are the where we actually go wrong. So here where we may go wrong is that we may go wrong between X and XXXO and XXXY types, right? What are the different examples you see in ZW type, and the difference between the XX and XY type, right? So when we see this, examples are important. XXXY, XY means two different gametes are forming, right? So here male gametes are two different. So that is human and Drosophila is example. Like that XXXO. Again here, what are the examples? Grasshoppers and many insects, right? And if you see the other type of expression systems, and their examples are more important and where we go wrong. Okay, so I'm not explaining each and every aspect. Thinking the scientist name is important here because he identified this X body, right? So these are the as for today, and again, uh, so that's the as of now, the next topics of this lesson would be covered in part 2, right? Part 2 of principles of inheritance and variation. We will start with this lesson tomorrow, and then we'll finish it quickly, and then we'll go for the next next lesson of botany, right? So that is what is given actually. Environmental issues we will start tomorrow. Okay, so this is it for today, guys, and then again I'm announcing that is I'm going to release a series of videos. In that, outline will be there, at the same time full chapter explanation will be there part wise. So if you see ecosystem, you will find two types of videos. One is 1 hour deep description or deep outline, and then one more type of video you will find in the same playlist, that is if you see part wise entire chapter. Okay, so these are the two types of videos you will be finding in one single playlist. Okay, likewise I will create the playlist according to the chapter wise. So each chapter you will find two videos, two types of, one is outline video and one is full video, I mean full chapters part wise. Okay, so please make use of it, and that is it for today, guys, and I hope this classes and my explanation and my our tips and tricks and discussion is really helping you out, and please leave the feedback in group as well, and please let your friends and share to other metric students, please, because metric students are they find it very difficult this time because they can't go to other institutions for getting coaching right now. This is an online platform is what they can make use of it. Be generous, and please share these videos to your metric friends, and let's have a healthy competition between everyone. Let's reach let these information and videos reach all the students. That's only my motto. Okay, please do subscribe and like this video before you leave. Okay, see you in the next class. Thank you. Stay home, stay safe.